we need to think a little bit about evolution again. So I put this quote, put that one in my microbiology classes in particular, because I think if you approach the content you're learning this semester in this class in the light of evolution, the evolutionary process, I think it starts to make a whole lot more sense, especially when we start talking about where microbes live and how they are capable of doing that. If you frame it in the evolutionary context, I think it does make it a little bit more intuitive. Now, what I'm going to be talking with you about is a little frustrating because they keep shifting the information because the information comes to light and you have to revise what was said before. So what you're seeing here is what was said before in terms of what they were thinking about Earth history, and then when we started to see signatures of life. So, for example, the first traces in biochemicals is what, 3.85 billion years ago. Some people talk about giga years, that's the same thing as billion years ago. So what was, first it was so oldest fossil 3.7 billion years ago. So in that area, roughly 3.7, they think that's when we started to see cellular life. Now they've changed it. Well, some people have proposed a change. I'll put it that way. This is published this summer in the journal Nature, Ecology, and Evolution, which is a big hitter journal. And so this has gone through a rigorous peer review process, which means other scientists in this field have rigorously looked through this and deemed it acceptable for publication. So there is more than just, you can see there's a lot of authors on this, which is kind of typical these days, by the way. Another thing pointing to this idea of interdisciplinary work. You start to see a lot more authors' papers. But here you can see, lots of authors, but it's gone through, other people have looked at this very rigorously. And so the nature of the last universal common ancestor, what does that abbreviate down to? Do you remember? LUCA, and its impact on the early Earth system. So now they're shifting it to 4.2 billion. So that's quite a bit earlier than they thought, which I've seen some other reports over the last couple of years in the literature, some published papers where they're hinting at this, but this is one of the most rigorous studies I've read. And I'll be honest with you, I'm not an evolutionary microbiologist, so some of it was above me in terms of what they, especially their methods, but it's, they referenced a lot of other papers to back up what they were stating. And then they did some very, very fancy work here in order to come to this conclusion. And what was actually quite surprising is that there's more to the genome than what they thought, a bigger genome than what they thought ancient life would have. And so 2.5 methyl-base pairs, 2,600 proteins being coded for. So they say that's comparable to modern day prokaryotes. And they think that LUCA was a prokaryote, an anaerobe, and a cetogen that actually possessed an early immune system. So that's pretty fancy. Previous studies were thinking that the earliest ancient cellular life was a little simpler than that. So it's an interesting proposal here of what they think is going on. So anyone remember or know what anaerobic means? Technology. Yeah, without oxygen. So that kind of makes sense, because when you look at all the information they've been able to gather about early Earth atmosphere, there wasn't oxygen. And so the earliest cellular life came into being without oxygen around them. And so they had to deal with that. So anaerobic. Acetogen, we're going to talk a little bit more about that later, but basically, gen, we see that on the end of a science word that means to create. Acetogen, acetogenesis, create acetate. And so they are using a pathway where acetate is involved. And we'll talk about that later. And they're saying, although LUCA is sometimes perceived as living in isolation, we infer that the LUCA to have been part of an established ecological system. So a member of a population. And LUCA would have provided a niche for other microbial community members, and hydrogen recycling by atmospheric photochemistry could have supported a modestly productive early ecosystem. So we'll get a little bit more into what all that means, but basically, they're saying that the LUCA was part of a community. There were interactions going on, and we're going to learn in this class, especially physiology, like how when microbes live in populations, which in natural systems, that's what they do. When they live in populations, communities, they very much are taking advantage of what others are doing around them, in the sense of if one microbe does its thing and excretes a certain waste product, another microbe nearby will go, oh, I like that, I'll take that and use it. That, what's that saying? Another person, one person's trash is another person's treasure. It's that idea. And so, and that also, once you start to learn that idea, it also helps you to figure out how these communities can sustain themselves and really have a nice niche in that particular environment. So this is from the paper where they're saying that, you know, it's a little bit more sophisticated cell than what they thought. And this is proposing about 4.2 billion years ago. I'm not going to get into the details of this other than to point out, you know, you can see there's stuff going on here. Okay, it's not an empty cell. They're proposing that, actually they're proposing that DNA is not there yet. If it was DNA, okay, this is going back to DI 111. If DNA, what would be one of the bases there that's not showing up? T, right? T would be the base. So they're saying strongly by this drawing that it's an RNA type molecule that's being used for information storage, which matches up with what other people. And then this is pointing to the idea of an immune system. And if you've heard about, you know, like CRISPR-Cas system that's being used in molecular biology all the time, and it's being used in some really amazing ways to help human health. And that's actually taken from microbes. So microbes do it. That's what they do, or many do. And it's a way for them to defend themselves against foreign DNA that comes into them, which we'll talk about soon here. And so you have this idea that, and they're also saying, yes, they were able to figure out a way to make ATP, which is our energy source of pretty much any living organism that we know of on Earth. So more sophisticated than we thought. So shift for, I think it's fair now, I read enough papers that we can shift from 3.7 to about 4.2 and start accepting that for now, but realizing when it comes to evolutionary research, it could shift again. So that's why I don't go into huge detail, because it's always evolving, right? So, you know, as they're studying all this, they're certainly thinking about what was the early Earth like, how did it differ from what we currently experience. Yeah, that's what evolutionary microbiology think. That viruses definitely. Yeah, like the more research going on with viruses these days, they're just everywhere, and including in our atmosphere. Yes. Yeah, some microbiologists propose that actually viruses came first before cellular life. because viruses aren't cellular. That's why I keep saying cellular life, because some people think viruses were there first, and that was kind of a prelude to cellular life. Dr. Sharp, who teaches virology and immunology, he may have some current research information on that in terms of the primary literature. But that's, some of the readings I do. I'm not a virus expert, but that's what many prominent virologists are saying. So certainly the atmosphere they propose is very different. The geology, when Earth was forming, I think you might remember from, basic evolution that we had supercontinents, and we don't have the continents spread out like what we do now. And when you look at the atmosphere back then, they proposed that it was rich in hydrogen gas. So that's that paper we were just looking at. They were mentioning hydrogen recycling. They knew that hydrogen was a pretty big deal. There's a lot of actually interesting research going on in hydrogen and human health. Has anyone heard of hydrogen water? I don't know much about it, but I've read a couple things and I'm like, huh. It's intriguing. Has anyone tried it? I have not tried it, but hydrogen is like H3O. Yeah, the hydrogen water, I looked into it. There's a couple different ways it can be produced safely. It's a safe thing. But kind of hold that thought, because hydrogen, H2, is one of the best ways that microbes capitalize on making ATP by using H2. And so a lot of health people are saying it could be good for us. Because remember, our mitochondria were once free living microbes. It's our mitochondria that's the powerhouse for ATP production. And they do it by producing a proton gradient. And you can get protons from H2. And so that's kind of the logic they're using, but I haven't looked into it deeply enough to know if there's any substance. But maybe some of the physiology professors, I should pick their brain, ask them about it. Anyways, hydrogen rich, CH4 methane, ammonia, of course, water, and CO2. So very different in the sense that there's no oxygen, no huge amounts of dinitrogen. And of course, early Earth chemically was developing. And you'll learn a little bit about that tomorrow when you watch that video with Dr. Lynn Margulis. She'll talk a little bit about that. So the famous experiments that were done to start thinking about early Earth and the evolutionary process and how cellular life came about was by Stanley Miller's work. I believe this was actually his master's project. And so Stanley Miller really kind of became famous for these types of experiments where in a regular chemistry lab with regular chemistry equipment and supplies, he was able to show if you provide the right types of conditions, you can take simple chemicals and end up with amino acids, simple amino acids. So you're going from, monomers, simple chemicals to polymers and more complicated molecules. And so you can see that, he did provide certain types of gases that they thought was common in the atmosphere, and then ran, with some heat source and looked at what they could get from this process after it cooked for a while. And glycine was the first thing that he was able to show was synthesized. And it's the simplest of the amino acids. And then further experiments showed other amino acids could be produced through these methods. So it was a huge breakthrough back then because I can't remember what decade this was, but it was showing that process that you can start with simple chemicals and end up with more complex. And so that started to support some of the other ideas that other scientists had in terms of, how did things progress to eventual cellular life? Going to nucleotides, that's a little bit more complex than just glycine, for example. But they could show that this could happen too under the right conditions. And it often has to do with having the right types of minerals in the environment to kind of serve as they often use what's the phrase? Kind of like making pancakes or crepes, where you start with simple batter and then you provide that heat source in it. Don't you love making pancakes? It's so magical. You know, you get that batter and then you put it on the skillet and it morphs into this nice, fluffy, delicious pancake. Kind of like, so the phrase is from soup to crepes. Crepes are fancy pancakes, which I don't know how to make those, but I know how to make pancakes. But soup to crepes is like the famous saying that came about. And so they had to, you know, ponder these ideas, use as much evidence as they could. Of course, the fossil evidence was playing a huge role in that, but it's pretty hard to find signatures of biochemicals in fossils. Again, my hat is off to those who study this. It's a daunting task. And so, you know, here we're starting to list out things that we find in life on Earth in cells. And so that soup to crepes idea, they think that a mineral surface is important to certain ways to catalyze these reactions. So this is a mock-up trying to show in this case how perhaps nucleotides were able to be synthesized under the right conditions. And so then there, you know, the other big idea is, where did cellular life arise on the planet? And now that's starting to come under debate again. That first paper I just showed you, the 2024 paper, they're starting to back off from what I learned, which is, and I'll talk to you about it, deep in the ocean. They're saying, yeah, maybe, but maybe not. So, but you have to find the right type of condition in terms of what they do. They're really starting to get this more solidified in the sense that, yeah, it seems like a mineral surface is really important for this process of polymerization into larger molecules. And so hydrothermal vents deep in the ocean floor are currently, that's what's being proposed as the site of cellular life being able to form. That 2024 paper is also saying, yeah, okay, that's plausible, and we're not ready to throw that idea out, but we need to also consider it could have been shallow ocean. And that's like way back when I was an undergrad, that's what I was, shallow ocean. And then it shifted to deep ocean when I was in grad school. And now they're going, maybe it's shallow ocean again. So again, it's a little frustrating, but basically we're looking at the idea of, you know, there's usually needs to be some kind of heat source and the right mixture of chemicals, of course, and then some kind of mineral surface. Pretty much everyone is agreeing that the RNA coming first before DNA is a thing. That's what they think happened. So RNA world, you might have heard that phrase. And some people say maybe it was a peptide nucleic acid mix that was actually first, and then the more RNA as we know it today came about. So that's still something that's out there I see in the literature. Peptide nucleic acid, they think it could have been a glycine amino acid and serving as a backbone. So that's your peptide idea, and then bases attached to that. And you know, Stanley Miller did show how easy it is to make glycine. So some people support that idea. Here they're just trying to show this idea in visual form, where you have the RNA molecule coming first. And then here they're showing this idea of how do we go from individual chemicals to cellular life? And then that has to, you have to start thinking about a phospholipid membrane for that. So here they're trying to show a lipid or protein vesicle. And I mean, it really is kind of hard to wrap your head around it, because you have to imagine the right environment with the right chemicals, the right mineral surface, and then you have all these chemicals floating around. They still think it's a watery environment where all this happened. So then you have to imagine a phospholipid vesicle surrounding those chemicals, and eventually the right mixture of chemicals were surrounded to start to become a functioning cellular organism. It's kind of hard to imagine, but that's kind of what they're getting at here with this image, that if you get the right mixture engulfed, so to speak, in this membrane, and it's all the right substances, then you can start to have an independent cellular organism capable of persisting and replicating. So it's kind of mind-blowing research. It's a really intense field of study, lots of unknowns, and it's shifting. Like I said, it's shifting quite a bit in terms of, with more knowledge, they start to update and kind of shift the story a little bit. But when you think about RNA coming first, I mean, that does make sense. RNA is simpler than DNA. And we know that reverse transcriptase, is an enzyme that can take information off of RNA and turn it into DNA. So they think that that could have been a way. And viruses, by the way, have many viruses, not all, but many viruses have reverse transcriptase. So that enzyme was there. And they think that it could have happened where that was being used to convert to DNA, and eventually the information storage molecule became DNA instead of RNA, mainly because it's more stable than RNA. And then you have to think about those lipids. If you've ever, I don't know if they do this in your chemistry classes at any point, but it's pretty easy to demonstrate. If you drop phospholipids into a beaker of water, you automatically see them form vesicles. Like they'll form a spherical vesicle, and it becomes a bilayer net, like a cell. And so it's just the way that phospholipids interact with water. Parts of it is very water friendly, hydrophilic, and wants to interact with water. Other parts of the phospholipid don't like water, phobic, away from it, and that creates this phospholipid membrane vesicle that could form. And again, in a watery environment like the ocean, you can imagine how that might have allowed for those vesicles to just spontaneously form under the right conditions. And if they enclose the right other molecules, you could then end up with some of them being primitive cells or protocells. And so for me, it's really hard to imagine. It's kind of funky to think about and fun to think about. But now they're saying this could have been happening about 4.2 billion years ago instead of 3.7. So let's say that happens, and it has. Then you have to go from a protocell that's pretty simple, although that paper is saying it's more sophisticated than thought. And you have to think about cells have to go through pathways to stay alive, physiological or biochemical pathways, in order to get ATP, get their carbon, be able to have enough to reproduce and make progeny. And so, they think it was a pretty step-by-step process. Although that paper we just talked about, they're saying, it seems like there's some cells that were pretty sophisticated right from the get-go. So maybe this happened faster than they thought in terms of geological time. But basically, this idea of developing pathways where, microbes or protocells were using, let's say, nutrient A, and that was their source of nutrient. But if their environment became depleted, then they'd have to adapt to taking in something else and converting it to nutrient A. And so you can start to see how there would be some pathways as long as the right enzymes could be evolved in the process here. And so really it's driven by their environment, how they're going to come up with these biochemical pathways. So keep this in mind as we get into some of the physiology in this class. It's really, when you look at microbes, are just astounding at the amazing variety of metabolic pathways that they have. The science fiction has nothing. Microbes leave science fiction in the dirt. And when it comes to how amazing they are, especially with their physiology. And if you keep in mind, it's driven by their habitats. What are they surrounded by? And microbes are amazing at adapting quickly to their environment. And so coming up with new enzymes isn't that hard for microbes compared to how, we don't really evolve and adapt very quickly at all to our habitat. Microbes can do it on the fly, and they're constantly evolving. And so here they're trying to show here how if nutrient A, the green balls, become depleted, then they'll start taking in another nutrient that's available to them. They create the enzyme through natural selection processes to convert it to A, then they're good. And again, the more complicated pathway on the bottom there showing nutrient C coming in, getting converted to B to A. So this idea of simple metabolic pathways becoming more sophisticated. Those of you who have had Gen Micro, this looks familiar. So this is often what they say could have been like one of the first pathways that microbes back then came up with in order to get their ATP. And so I like to introduce this concept because there's a couple of key things here that is going to be kind of a theme running through the semester as we talk about microbes and how they survive in their habitats. But here, if you look at it, you can see there's no oxygen. So they're talking primitive, ancient life, no oxygen in the atmosphere. So up top is what they're showing could be going on in the atmosphere, right? Where you have ultraviolet light playing a role. And they're saying, if the sun is shining and you have iron, Fe2+, and a proton, they'll spontaneously react under ultraviolet light to become hydrogen gas. And then Fe3+ is ferric. That's the ferric form of iron. I would like you to know that because we're going to talk about iron a fair bit in this class. So the ferric form of iron, the Fe2+ is the ferrous form of iron. So I'd like you to know that, please, because we're going to get into iron use in lab, and we'll talk about it in lecture. So ferric and ferrous forms of iron. But you see here that you have spontaneously in the atmosphere hydrogen gas being created. That's a good thing. You also see they're showing another way to create some hydrogen gas. Whoops. Which is that reaction closer to the cell here, where you're looking right here. But again, if you look at the simple reaction, it's an iron sulfide reacting with H2S, by the way, that's a gas, that's hydrogen sulfide, it's a gas. That's the thing that smells like rotten eggs. If you've ever gone canoeing or kayaking and you put your paddle down deep into the sediments or silt and you lift up really quickly, you see all those bubbles coming up, and you might smell rotten eggs. That's microbes in silt producing hydrogen sulfide. So anyways, you have those reacting. And again, one of the major products here that is a really good benefit is the dihydrogen. So hydrogen gas is really good because what you see happening here is the use of some primitive hydrogenases that are in the cell. So what they're showing here is a phospholipid membrane with some primitive enzymes embedded in the membrane. And then the lower portion of the figure is the inside of the protocell, the ancient microbe. So one of the key things happening here is you see this primitive hydrogenase is acting on dihydrogen. And what it's doing is it wants the electrons. So it's stripping electrons off of hydrogen gas. You have to remember that hydrogen has one proton and one electron. So if you strip an electron off of it, you're left with a proton. So there you see the proton, okay? You see that electron getting fed into the cell, and it's being used for this reaction in the cell, which is water, S to the 0. Anytime you see to the 0 like that, basic element. So anyways, it's the electron comes in and facilitates completion of the reaction, and look what you end up with. You have hydrogen sulfide gas. And then they show the dashed line, and they say that gets excreted out of the cell, which then facilitates that reaction. So it's kind of sweet. If the microbe can do that. It's then recycling that out. Well, that reaction can happen again, so this can happen again. And as this is happening, so as all of that left portion is happening, you have this buildup. So as this is happening over and over and over and over and over again, you have to imagine you're going to cause a buildup of protons, right? Because that's one of the products. As you strip electrons off hydrogen gas, you have protons. So I wish the artist would have been a little bit more generous. Lots of protons show up. So if you have lots of protons on one side of the membrane, less protons on the other side of the membrane, you have a proton gradient. And that's going to become a theme in this class. You got to start thinking about proton gradients now, because that is potential energy. That is a good situation for the micro because it can use a proton gradient to accomplish certain things. What is it using it for in this particular case? Yeah. And many, many, many organisms do this all the time. You're doing it right now in your mitochondria, especially if you have good breakfast. So you have this proton gradient, and they're showing the protons are going through this primitive ATPase, which is an amazing enzyme. It is, they refer to it as a molecular motor because it actually has subunits that rotate and create a torque force. And as the protons are moving through it, that's what's actually causing the torque force, which allows the organic phosphate to come into contact and react, and then you get ATP. So this whole little, this is a pretty simple pathway. There's actually a couple here that are linked together by that kind of recycling of hydrogen sulfide. And as long as they can keep that going, creates that proton gradient, which is a pretty simple way to make sure they have enough ATP. Questions on this concept? Yeah, I think there were some upgrades in the subunits, but it's a good question. I don't have an answer to, but I get the impression, especially that 2024 paper I was reading that I talked to you a little bit about earlier. I think in there they talk a little bit about the subunits of ATP and how it might have differed to current ATP. So yeah, they are showing it as a pink blob, but there's lots of subunits that are involved in this. So. Did you ever, if you took BI 111 here with Dr. Lindsay, did you ever act out ATP? Early on, when he would have students do that years ago, where they'd have to get up and students would be the subunits, and then someone would be the protons, and it creates like this little dance as the subunits are moving. It's a good idea. And by the way, hydrogen sulfide is a gas and it's toxic. So that's another reason why they're like, yeah, I don't want that. So that's, they're getting rid of it. It's a waste product. But it's a very useful waste product in the sense that it's allowing for that other reaction to keep going. Make sure that it's a lovely hydrogen gas being synthesized. Any other questions on this idea? So here you have a phylogenetic tree. I couldn't get the loop out of the circle. Sorry. It wouldn't let me. This picture came from the textbook company. I didn't have control on the fine details of it. And it came offset like that. So this should be just the same. The black line is. So that's the LUCA. This is a very abbreviated phylogenetic tree coming from the last universal common ancestor. I would like you to know the 3 domains that are in the colored boxes. So those are at the domain level. So you have domain bacteria, domain archaea, and domain eukarya. Currently, that's how they're working the classification, where you have a domain level above the kingdom. A couple of things on here I want you to pay particular attention to is that when you look off to the blue on the left there with domain bacteria, you see, where is it? Oh, here. Look at that, mitochondria, chloroplast. So again, don't forget, the endosymbiosis theory states that those are once free living ancient cells that became engulfed into larger cells to form what we then call eukaryotic cells. So the domain bacteria and archaea are prokaryotic. They're no nucleus. Typically, when you look up the definition of prokaryotic, they'll say no nucleus and no other organelles. That part of the definition may need to be modified because actually they've shown that some bacteria and archaea do have what look to be like organelles, but so far no one's saying that word, especially in writing, because it's such a fundamental paradigm. I don't think they want to go there yet, but I'll show you some examples of that in this class. The other thing to be aware of when you look at this phylogenetic tree, if you look at the base here and then work your way off to the right, you end up eventually with domain eukarya. And on that line is where you see the archaea starting to come off. Okay? So the way to interpret that is that the archaea are on the line leading off to the eukarya. The archaea are actually more closely related to the eukarya domain of life than they are to the bacterial domain of life. That was a surprise. They did not expect that. But the DNA information coming out, DNA sequencing, it's supporting this more and more and more. So when I was an undergrad, archaea and bacteria were lumped into one group, that they were all the same-ish. And then the eukarya were very different. And then some very pivotal research was done where with the greater technology, the better technology with ribosomal RNA sequencing going on, they could show, no, we were wrong. Archaea actually are more closely related to the eukarya, even though they're prokaryotic. But yeah, so that's, we'll talk a little bit more about them as well. Archaea, you often get taught that archaea are very live in very hostile environments, which is true. Like you can find them in volcanoes. You can find them in deep hydrothermal vents. You can find them in the hot springs out west, where if you fell into those hot springs, you'd die. You can find them in radioactive water and nuclear power plants. So you can find these organisms, yes, in very hostile environments, but you can find them out there in the soil. They're out there. They love your compost pile. If you have a compost pile, you've made some good friends with the archaea. So they love that because the heat. A lot of them are very well adapted to high heat. I'm sure you might have noticed compost piles, good compost piles steaming. So you gotta imagine there's plenty of archaea there. But you can find archaea, even in the soil here. They're there. So they have a good reputation for hostile environments that they can adapt to. And we'll talk about that, but they're in other places. So here's some fossils pieces, and this is what they're finding with scanning electron microscope. And if you've not had a microbiology class, this may look like a bunch of nothing, but if you've had micro, these should look vaguely familiar to what you've seen microscope. So these are rod-shaped organisms, and much of microbial life is rod-shaped. And so how they find these needle in a haystack pieces of evidence, but they do. So again, very tedious. I would imagine lots of luck involved in getting this confirmed. So this is another piece of evidence that they look at pretty closely and have been studying for a long time. these are different images of stromatolites. So I talk about this in micro 2. So stromatolites, stop it. Stromato, L-I-T-E-S. Stromatolites. Boy. Stromatolites. Can you make sense of that? These are found, a lot of them that have been studied were in, are in Australia. There's current stromatolites that are current living stromatolites, organisms that are happily living in these consortiums. There's some fossilized stromatolites. So they stretch back for, I think the figure A is the oldest stromatolite that they have, fossilized. So the figure A is a fossilized one. But like these are current ones. You can level high, walk amongst these. And what it is layers upon layers or mats, layered mats of microbes, mostly photosynthetic cyanobacteria, but others in there too. And over time, you know, there's some that have become fossilized, they're so old. And so what they do is they take slices of them and study them, and that's where, you know, they start to get images like that, where you can see cellular life in those fossils. Here, this is one that was dated to 3.5 billion years ago. And the arrows are kind of showing that layering, or the mats that are laid on top of each other. This is in Australia. So those are probably, Australia stromatolites, probably the best study, but I was really surprised to find that we have some here in the Upper Peninsula. So this is a stromatolite, Precambium stromatolite, and cyanobacteria. Does it say here exactly where? Chocolate group. Here, coming from the Chocolate group in an area known as the Chocolate Hills, located near Ishpeming. That's pretty cool. And polished up to look quite beautiful with all those banding colors that you see there. The layers of, yeah, the mats of stone. So by the way, cyanobacteria is the term I want you to know, and I want you to advocate for this term out to your friends and anyone who will listen to you, because my pet peeve, one of my pet peeves, you know what other term they use for this? Cyanobacteria? No. Blue-green algae. Oh my goodness. It's not algae. Algae are eukaryotic organisms. Cyanobacteria are bacteria. They're prokaryotic organisms. So if you hear someone say blue-green algae, no, no, no. They're cyanobacteria, actually bacteria. So yeah. Isn't that beautiful though? So we have them here around Lake Superior area. So I'm sure they'll even find more. They keep looking for them. So yeah, the stromatolites have been a wealth of information in terms of, you know, the fossilized ones in particular. But remember, there's current ones too. Those are not all fossils. There's current stromatolites. If you go to Australia, you can certainly walk amongst them if you want to. All right, we'll stop there. We're going to get into a little bit, just a little bit more on evolution, and then we'll start looking at some physiology.